Hello, Junior Lab Rats. We're going to go ahead and roll through our second lesson here today, which is about forming a hypothesis and how we use the scientific method. All right, so remember to keep your notes out, follow along, and if you need to pause at any time, go right ahead. So here is some of your vocabulary from the very beginning of your notes in your chapter. So your words are scientific method. I'm going to read through these with you, but we're going to make more sense of them as we go along, okay? Scientific method, a systematic method, which is a series of steps, including observation, forming a hypothesis, experimentation, and data analysis. A hypothesis is a proposed explanation for an observation. It's a prediction or an explanation that is tested by an experiment. It is a tentative answer to a question. In science, a hypothesis is part of the scientific method. Observation, that's receiving knowledge of the outside world through our senses or recording information using scientific tools and instruments. Experimentation, a scientific procedure undertaken to make a discovery, test a hypothesis, or demonstrate a known fact. Data analysis is the detailed examination of facts and statistics collected. Conclusion, an interpretation of or a judgment or decision reached by reasoning and data analysis. To repeat something is to do the same thing again. To replicate means to reproduce, right? So if you replicate your results in an experiment, you reproduce them. That's what this is going to mean. And to revise means to re-examine and to make changes. Now, these last two definitions, independent or manipulated variable and dependent or responding variable. Okay, so you're going to understand these more as you have more experience with them. But also, the terminology gets a little confusing. So I believe the Lab Rats textbook uses the term manipulated variable and yours uses the term independent variable. They are the same thing, but more often than not, I think independent variable is used more commonly, and it's typically the term that falls out of my mouth. Dependent variable, again, I think is used more commonly, and the other textbook for the Lab Rats class, not the junior class, uses the word responding variable. An independent variable can be manipulated by an experimenter and can cause a change in another variable. A dependent variable would be can change if other factors change and can change in response to a manipulated variable. So I know this is a little confusing, but I'm going to give you an example of some variables here in a proposed experiment, and we're going to keep coming back to this example during this lesson. So say you have a headache and you want to know what fixes your headache, gets rid of it. So you want to try a couple things. So you want to try peppermint oil, like smelling peppermint oil. You want to try Tylenol. You want to try putting your feet in ice water, and you want to try sitting in a dark room. All right. Those would all be independent variables. They're the things that you're going to change to try. And your dependent variable is going to be, for example, your headache. Like, does it get better? Does it get worse? Okay. Now we're going to talk about how we use those variables and how we experiment with them as we continue. Okay, so what's a hypothesis? We're going to break down that scientific method terminology that was right up here, this first one. See how it has all these different words in it, a hypothesis, experimentation, and they're all here. We're going to break them down even more. So what's a hypothesis? It's a proposed explanation for an observation. In science, a hypothesis is part of the scientific method. It's a prediction or an explanation that can be tested by an experiment. Observations and experiments may disprove a scientific hypothesis, but can never entirely prove one. Okay, I believe your book may have used the word prove. One of the textbooks did. It's not really good to use prove or disprove. Really, the better terminology is support or not support. And I'm going to mention that a couple times in this lesson. I probably should have gone back through, but I didn't realize that I still had this in here when I started recording. Most scientific hypotheses is plural of hypothesis, are proposed in an if-then format because it's easy to design an experiment to see whether or not a cause and effect relationship exists between an independent variable and a dependent variable. The hypothesis is written as a prediction of the outcome of the experiment. So it's what you predict will be the outcome of your experiment. And it's very often if-then. It's not always if-then. So here are some examples. So here's an if-then example. If you drop a rock and a feather, they will fall at the same rate. Now, it doesn't say the word then, but if you drop a rock and a feather, then they will fall at the same rate. That's an if-then statement, okay? This is not an if-then. Plants need sunlight in order to live. Now, you could test that, but it's not if-then, okay? Eating sugar gives you energy. There's another and that actually is an if-then, because you could rephrase this: if you eat sugar, then you will have more energy, for example. So a hypothesis has to be testable in order for it to be something you can experiment with. So what makes it testable? A hypothesis is a tentative answer to a scientific question. Tentative means it sort of means it's like your current answer. Like, say you're invited to your friend's house on Friday night and you're thinking you can probably go, but you're not sure. You might say, my tentative answer is yes. It means, yeah, I'm thinking that's right, but I'm not 100% positive. A testable hypothesis is a hypothesis that can be, this should say, really supported or not supported instead of proved or disproved as a result of testing, data collection, or experience. Okay, so it could come out as supported or not supported, right? Only testable hypotheses can be used to conceive and perform an experiment using scientific method. So what does a hypothesis need to be testable? The requirements would be, in order to be a testable hypothesis, 2 criteria must be met. It must be possible to prove, or we should say support, if we're going to use better terminology, support that it's true, or we must be able to really support that it's false, or at least say it can't be proven as true, or it can't be supported as true. It must be possible to reproduce the results of a hypothesis. That matters. If you can't reproduce the results of your hypothesis and experiment, then you can't say that it wasn't a fluke, that the next 5 times you do the experiment, you're not going to get a different result. We're not going to have time to repeat most of our experiments, but what we do have is 4 lab groups. So essentially, every group should be getting the same results if everybody is conducting the experiment the same way. So here are some examples of a testable hypothesis. Students who attend class have higher grades than students who skip class. This is testable because it's possible to compare the grades of students who do and do not skip class, and then analyze the resulting data. Another person could conduct the same research and come up with the same results. Here's another testable one. People exposed to high levels of ultraviolet light have a higher incidence of cancer than the norm. That would mean than your normal risk, okay? This is testable because it's possible to find a group of people who've been exposed to high levels of ultraviolet light and compare their cancer rates to the average. So perhaps what you would do is find a group of people who work outside versus a group of people who work inside, right? Find out who is exposed to more ultraviolet light. If you put people in a dark room, then they will be unable to tell when an infrared light turns on. This hypothesis is testable because it is possible to put a group of people in a dark room, turn on an infrared light, and ask the people in the room whether or not an infrared light has been turned on. So those are all good examples, testable hypotheses. These are all good hypotheses. These are not testable. These are not great hypotheses. These are not the kind of hypotheses we want to be making for our experiments. It doesn't matter whether or not you skip class. This hypothesis can't be tested because it doesn't make any actual claim regarding the outcome of skipping class. What does "it doesn't matter" mean? What does that mean? It doesn't have any specific meaning, so it can't be tested. Ultraviolet light could cause cancer. We don't want the word "could" anywhere. It makes a hypothesis extremely difficult to test because it's very vague. The "could" here, for example, there could be UFOs watching us at every moment, but it's impossible to prove that they're there, so could's not good. We don't want to say "could" in anything. How about goldfish make better pets than guinea pigs? That's not a hypothesis. What is it? It's an opinion, right? There's no agreed upon definition of what a better pet is. So while it's possible to argue the point, you can't test it, you can't prove it, support it or unsupported it, because it's entirely about preference and opinion. A hypothesis is a tentative answer to a scientific question. A testable hypothesis is a hypothesis that can be proved or disproved or supported or unsupported as a result of testing, data collection, or experience. Only testable hypotheses can be used to conceive and perform an experiment using the scientific method. So this means come up with, conceive, and perform, right? So if you have been watching and not filling in blanks in your notes or looking over your notes, you may want to stop for a second and check it out. Make sure you haven't missed anything, because we're going to move on now to what is scientific method. All right, so scientific method is defined as a series of steps to collect information or solve problems. So I want you to notice this is a nice, neat little line, right? I'm going to show you in a minute why it doesn't really look like this. Okay, this is just oversimplified to give you an idea of the parts involved. So you've got observation, hypothesis, experimentation, data analysis, and conclusion. So it says, ask a question, do background research, construct a hypothesis, test your hypothesis by doing an experiment, analyze your data and draw a conclusion, and then, report your results. Was your hypothesis correct? All right, these steps do not always go in this order. They don't always stay in a nice straight line. Far more often it looks like a big tangle, and you'll see why in a minute. But it's very important that you follow these steps and that you maintain a scientific method during your experiment. And this part about testing your hypothesis, this is where variables come in, and we're going to talk about that more in a minute. So we have your observations. Those are things like taste, hearing, sight, smell, touch, your formation of a hypothesis, your experimentation, your data analysis, and your conclusion. And those are usually parts of a lab too. So what's observation? So observation consists of receiving knowledge from the outside world through our senses or recording information using scientific tools and instruments. The data recorded during an observation can be called, during an experiment can be called an observation. So yes, you can taste, hear, see, smell, and touch things. There's also a vestibular sense that we don't usually think about when we think about our five senses, because we do have more than five. For example, if you're in a car and you close your eyes, can you tell which direction you're moving? Left, right, straight, backward, up a hill, down a hill? That's a vestibular sense. And usually it's not mentioned when we discuss senses, but it's very important. And also write, what was I going to say? Okay, there's that. So tools, that's what I was going to say. My mind went blank for a second. Tools, so things like scales and thermometers, okay? So a hypothesis, again, remember we just said it's the initial building block of a scientific method, and many describe it as an educated guess. It's based on prior knowledge and observation. So while it's true, the definition can be expanded. We went through several pages of what a hypothesis is. So you guys should have a decent idea now. What's your experiment? All right, scientists and other people test hypotheses by conducting experiments. The purpose of an experiment is to determine whether observations of the real world agree with or conflict with predictions derived from a hypothesis. So it says right here, agree with or conflict with, and that is pretty much support and do not support, or the less favored language, the prove or disprove, which really isn't the best language. So look, they have an experiment. I believe they have a distillation kit here, and that's super cool. Data analysis. So generally speaking, scientific data analysis usually involves one or more of the following three tasks: generating tables, converting data into graphs or other visual displays, and or using statistical tests. All right, now it doesn't always involve these things, but I will tell you, when you convert your data into a graph or when you use a table, it is usually far more often to understand it, far more easy to understand it, because often it gives you a visual representation of the data you collected. So if you do a graph involving the height of a plant and if you just write down your measurements, that's one thing. But if you do a graph and you show that it gets bigger and bigger every day, it's just much easier to understand your data. So conclusion. Scientists draw conclusions by examining the data from their experiment. There are basically two possible outcomes. Either the experiment supported the hypothesis and it can be regarded as true or supported, basically, or the experiment disproved a hypothesis and the hypothesis is unsupported or false, right? Oops, this guy's mother. And if your hypothesis is supported or proven true is what we were thinking about before, but this means it's supported, make sure you can repeat your process and replicate your results. One of the core principles of scientific process is that other scientists are able to repeat your experiment and either confirm or refute your results. This is referred to as reproducibility or replication. This is important. If you only do it once and you get a result and you say, yes, this is absolutely, my hypothesis is correct, it's supported. How do you know that there wasn't a mistake somewhere along the way and your hypothesis isn't really supported based on how you initially wrote it, that whatever you did in your experiment, you measured wrong or you boiled something too long or, whatever. You have to be able to replicate it to show that it is always supported or most often supported. If your hypothesis is not supported, oh, and by the way, that's what I mean by it can be proven false or unsupported, but you can't ever really totally say that it's completely supported, because there may be times when you get a different answer and you can't explain why. So if your hypothesis is not supported, revise it. So sometimes it's a matter of just repeating the experiment exactly the way you did it. Other times you should revise your hypothesis. Go back and look at what you should change and see if you can change something. Maybe you boil your solution a little bit longer or add a little more of this or that chemical. Sometimes it's just, I'm not sure I did it right, so I'm going to repeat it exactly the way I did it. That's why we have 4 lab groups, because everybody should be doing it the same way, right? So keep calm and revise if you need to. So really, your scientific method looks more like this. That's not a nice, neat, straight chart, is it? Here's where we started with the ask a question, do background research, construct a hypothesis, test with an experiment. But then you get to this point. Is your procedure working? No. Troubleshoot it. Carefully check all the steps and setup. Go back and do it again. Is it still not working? Well, how many times are you going to do this before you revise? At some point, you're going to revise. Well, what if it is working? Okay, if it is working, then analyze data and draw conclusions. If your results align partially or not at all with your hypothesis, you can come back up here and it says experimental data becomes background research for new and future project. Ask a new question, form a new hypothesis, experiment again, and then you go through these steps again. But what if it does align? Then you can communicate your results, but you also should repeat it as well, just to make sure that it is able to be replicated. But that's why we have 4 lab groups. So types of variables. So we're going to talk here for a minute. Earlier I said, imagine an experiment where you're trying to figure out what's the best way to get rid of a headache. Sticking your feet in ice, sniffing peppermint oil, sitting in a dark room, or taking a Tylenol. Okay, those are all variables. And those are all actually independent variables. That's the thing you change. So I want you to imagine that you have a headache and you do all 4 of those things. You go in a dark room, you bring yourself a cotton ball full of peppermint oil to sniff, you take a Tylenol, and you stick your feet in a bucket of ice water. Okay, your headache gets better. How do you know which one did it? Was it the ice water? Maybe it was the Tylenol. You don't know, because you have too many variables. You should really only have the one. It says right here, the one thing you change. Limit to only one in an experiment. So if you want to know if those things will help make a headache go away, you have to try one every time you get a headache, and only one, and change nothing else, because that is how you determine whether or not that particular variable contributed to your result. You don't know if you do all 4 at the same time. Maybe it was only one of them. You don't know. So your dependent variable is the change that happens because of the independent variable. So this would be your headache. Does it go away? How quickly does it go away? Does it get worse? And then your control. That's everything else that you would want to remain constant and unchanging. So in our example, it would be like the temperature of the room you're sitting in, the clothes you're wearing, your level of activity, whether or not you've eaten, what was the last time you ate, how much sleep you've had. All those things might contribute. So in these examples, their independent variable is the liquid used to water each plant. The dependent variable is the height. And the control is the type of plant used, the pot size, the amount of liquid, the amount of soil. Okay? So they're watering plants with like water, juice, and soda. I don't know. So your goal this year is to be a good scientist. And these are 2 of my favorite scientists. This is Bunsen, if you don't know him, and this is Beaker. and they are Muppets, and they are amazing. Beaker is a little terrifying in the laboratory. All right, you guys, see you next Tuesday.